today we're going to be talking about cell division. And when we talk about cell division, there's two types of cell division. We have what's called mitosis and we have what's called meiosis. Mitosis is going to involve what's called somatic cells. And meiosis is what's going to involve what's called sex cells. Somatic cells are our typical cells like a skin cell, a liver cell. When you think of most cells in your body, they're somatic cells. Now, sex cells, these are going to be specifically an egg in a female and a sperm in a male. I sometimes call these gametes there. And so meiosis is a separate video from this video here. This video will focus specifically on mitosis. So when we talk about cell division, we're going to be replicating our cells. We're going to be taking what's called a parent cell. So we're going to have this parent cell. I'm going to put a P right there. And we're going to be making two daughter cells. We're going to be making two daughter cells. And these two daughter cells will be exact replicas of the parent cell, which is very different from meiosis, because in meiosis, they're going to be having cells that will be, so what to say, daughter cells, but they're going to be making cells that are not replicas of the parent cell. And so when this parent cell makes these daughter cells here, the parent cell basically gets kind of engulfed into the two daughter cells there. So the parent cell disappears. Now the parent cell has 46 chromosomes. The two daughter cells will also have 46 chromosomes. This can also be stated as 23 pairs. So like I said, the daughter cells are going to be exact replicas of the parent cell, and this is specifically for mitosis. Meiosis will be different. And when I mention these chromosomes here, when I mention the term 23 pairs, from your parents, you get one chromosome from each parent. So you have this set of chromosomes. You have one from mom and one from dad. We'll talk about the structure of chromosomes in a little bit, but on chromosomes, there is segments of DNA, okay? And basically a gene is a specific segment of DNA. So chromosomes carry your genes, and your genes are specific segments of DNA, okay? Chromosomes, where are they located? Chromosomes are located in the nucleus, okay? And in your nucleus, so here's your cell. Remember, your nucleus is in the center of the cell there. The DNA, when it's not going through cell division, it's sort of spread out over the place as what's called chromatin, okay? But when we go through cell division, it will be compressed into these structures called chromosomes. And so you can see you've got one from mom and one from dad, and on these chromosomes are going to be specific segments of DNA, which would be specific genes, okay? There is some proteins that help pack these chromosomes together. These are called histones. Okay, so let's go ahead and let's get right into mitosis. Let's spend our time here talking specifically about mitosis. With mitosis, just to refresh your memory, you're looking at somatic cells that are going to be using mitosis, okay? They have 23 pairs. Okay, or we can say, let me clear that off there, or we can say 46 chromosomes in total. So 23 pairs or 46 chromosomes in total. So you basically get one set of 23 from each parent, all right? Now, when these chromosomes are sort of put together like this, let's say this one's from dad, and this one is from mom, all right? They call the term homologous chromosomes. So they call these homologous chromosomes, okay? Because they both carry the same thing. So for example, if this chromosome has eye color, on this side we would also have eye color. If right here we have attached earlobes, this one may have attached to it. Now, there can be different versions though. Like for eye color, let's say this one, the eye color is brown. Let's say over here it is blue. That's something we'll discuss in another chapter. For the earlobes, it could be attached over here, and this one could be unattached over here. So you can have different versions of genes, but earlobe attachment or unattachment is on this chromosome there. So it's the same chromosome for these pairs, these homologous sets of chromosomes here. And when you have a cell that has these paired chromosomes, the cell is said to be what's called diploid. All right, and so I'm going to put in parentheses 2n, okay? And so the 2n, that represents that you have a set of chromosomes. So you have one here and you have one here. You have paired sets of chromosomes. In meiosis, we'll hear a term called haploid, and that means that you would only have maybe one of them. So if you were a haploid cell, you'd maybe only have one chromosome of that pair, and that's something that we'll see in meiosis. Okay, so with mitosis, we have something that's called the cell cycle, and this is basically a set of events that the cell goes through actually prior to mitosis. So mitosis is cell division, right? Mitosis is cell division. Well, there is a prep stage prior to cell division called interphase. So let's just actually write this down, the cell cycle, and I'll show you a nice picture from a textbook source there. And so we have the cell cycle, right? You have what's called interphase, and then you have what's called mitosis, or the cell dividing phase, the cell division phase. This mitosis will further get divided into prophase, metaphase, anaphase, and telophase. And I know I abbreviated that, but believe me, we'll go through each of those phases individually. So let me show you guys a picture here. You can see that we have this image here. You can see that most of the cell cycle here is spent in interphase. You can see here that we have a little bit of time here actually spent in the mitotic phases there. Mitosis is specifically these prophase, metaphase, anaphase, and telophase. There is something at the end called cytokinesis, and that's going to be the actual process of splitting the cytoplasm. All right. So let's first start off talking about interphase. And we talk about interphase for our purposes. We're going to be talking about three subphases of interphase. And those three subphases is the G1, the S, and the G2. Okay, you can get even into more detail with higher levels of biology, but we're just going to stick with the basics here. And like I said, the cell spends most of its time in interphase. It's the preparatory stage. So if you think about it, if we have one parent cell and we're going to have two daughter cells, we're going to need two Golgi apparatuses. We're going to need two mitochondria. So if you remember all the organelles that make up the cell, we're going to need multiple copies of those organelles. Okay, and so with the G1 phase, this is going to be for growth and development. So a lot of the G1 phase is going to be doubling of your organelles, okay, doubling of the cytoplasm. So everything's kind of getting larger. So you're seeing rapid growth, increase in cytoplasm. Now, the S phase, this is probably the most important of the three phases, and this is where your DNA is replicated. And so when you say replicated, it basically means like we're going to make another copy of it. And when you make a copy of those chromosomes, they are called chromatids. So let me just show you an example of that down below. So remember, we have one from mom and we have one for dad. And you know what? I'm going to change the color on this and get real fancy here. All right, so let me, okay, so we have one from mom and we have one from dad there. And so what's going to happen is when we replicate the DNA, this is going to go like that, okay? And then let me give it back to my color there. And this one is going to go like that, okay? And so now we have a copy of mom's chromosome, and now we have a copy of dad's chromosome. And these are going to be attached at a certain structure at the center. And the structure is the same for the both of them. And that structure, these chromatids, are attached at that center structure called a centromere. All right, so these right here, so when the DNA is replicated and the two copies of the chromosomes are called chromatids, they are attached to the centromere. So these are usually they usually call them what's called sister chromatids. And I'm just going to write it over here. So when we make a copy of moms and dads. They're called sister chromatids there. Okay? All right, and so we get back to the G2 phase. This is where you start getting some cytoskeletal type of structural events. So like filaments, kind of like the framework or like scaffolding of the cell as it prepares for division there. Okay, so now that we're ready to go, there's actually an important component right here. There's an important component right after this G2 phase, and this is going to be called a checkpoint. Okay, and with these checkpoints here, this is probably your most important checkpoint after the G2 phase right there. And this checkpoint is going to basically say like, hey, did we copy the DNA correctly? Is all the organelles there, are they all duplicated? And the reason for that is because if the DNA is not copied correctly, we could potentially have a faulty cell, a crappy cell. And so we don't want that, right? So this checkpoint, if things fail, okay, if things aren't copied correctly, if the DNA is not copied correctly, then the cell would undergo what's called cell lysis. The cell would be called cell lysis here. Okay, so now we're going to go ahead and we're going to go into mitosis now. So now we're going to get actually into the phases of mitosis. And before we move on to this mitosis, let's just kind of review here. Let me get a different color pen here. Okay, so let me clear this off here. My eraser's not working. I'm just going to clear all. Okay. All right, let's try that again here. Okay, so with mitosis, you're going to have 4 phases. You're going to have what's called prophase. You're going to have what's called metaphase. You're going to have what's called anaphase. And you're going to have what's called telophase. All right, so I always remember it PMAT. And cytokinesis is a process that will occur after telophase. All right, so in prophase, if you look at some images, you will sometimes see that there is an early and a late prophase, okay? And so one of the big differences between the two is if the nuclear membrane is still present or if it has been fully broken down. So in early prophase, remember your chromosomes that were just replicated, your DNA that was just replicated, okay? That is in the nucleus, and we need to make 2 daughter cells. So we need to get those chromosomes out of that nucleus, correct? So we need to break down that nuclear membrane. So the nuclear membrane will start to break down, and sometimes they call this the nuclear envelope. That would be the same thing. Whereas in the late stage of prophase, the nuclear membrane is gone. So the nuclear membrane is totally broken down. Okay, we are going to be forming what's called a mitotic spindle. So that will start to form in early and will be completed in the late. Okay. Okay, so nuclear membrane breaks down, the mitotic spindle has formed here, all right? All right, so now we move into metaphase, okay? And the hallmark of metaphase is that the chromosomes will line up on the center of the cell. The chromosomes will line up on the center of the cell. Now remember, I'm trying not to get too fancy here. Remember, we have copies of moms and we have copies of dads, right? So it's looking something like this. Okay, so all these chromosomes here, they are lining up on the center of that cell. So they sometimes describe it as lining up on the equator of the cell. So if you guys know what the equator is on the earth there, so they sometimes reference that. All right, now I had mentioned something called a mitotic spindle up front here. So this mitotic spindle, I'm going to change the color over here just to make it fancy. The mitotic spindle is something that forms over here on the cell there. Okay, and it has these microtubules that sprout out. It has these microtubules that sprout out. So the mitotic spindle, it forms these microtubules that sort of sprout out, and they sprout out and attach at the centromere. They sprout out and attach at the centromere. If you remember the centromere was that little proteinaceous structure that had connected the two sister chromatids together. Now remember, we are going to have two separate cells, okay, with the same amount of chromosomes each. Remember, we doubled up all those chromosomes, right? So mitotic spindle, microtubules sprout out. I'm just going to add some notes here too. You have your chromosomes lined up at the center. Okay, now in anaphase, the hallmark of anaphase is this mitotic spindle with these microtubules is going to pull, it's going to pull these chromosomes to opposite ends of the cell there. So the chromosomes are going to break. They're going to break, and I'm just going to kind of spread this out as best as possible here. The chromosomes are going to break at the centromere. Okay, and they are going to migrate to opposite ends of the cell. Sometimes they say to opposite poles of the cell. All right, and so as we get to our last phase right here, we have what's called telophase. And let me draw that a little bit bigger here. I'm going to make it a little bit bigger over here. We have what's called telophase. You're going to get reestablishment of the nuclear membrane. Remember, we're going to have two daughter cells at the end, so we're going to have two nuclei. All right, and then these chromosomes that are in here, I'm just going to I should have saved that color there. Okay, these chromosomes that are in there, they're going to join with the one from mom and one from dad, like we said previously. All right, and what's going to happen here is you're going to get the nuclear membrane is going to be rebuilt. And I'm going to put 2 in parentheses because we have 2 new. If you were to completely stop this process at telophase, you would have one cell with 2 nuclei and a boatload of chromosomes there. So the nuclear membrane rebuilt, the chromatids or the chromosomes unite. Okay, and so what's going to happen after telophase, I'm just going to use this right here, is we're going to split the cytoplasm. And that's what cytokinesis is. So cytokinesis is technically not part of telophase. It's a separate event of mitosis, okay? And so the cytokinesis is the splitting of the cytoplasm. You can see it start towards the end of telophase. So you can see on a microscope when looking at this, you can sometimes see a little slit starting to form between the 2 nuclei. And cytokinesis is a result of microfilaments, which are tiny structural elements of a cell that sort of like pinch that cell down the center there. Okay, so you do in your notes have some other things that we talk about, but this is just the basis of mitosis here. And I want to just show you an image here really quick. And just going through an image here. Looking right here, actually this was not the image I wanted to show you. Sorry about this. Okay, so this is just showing you an overview before we end this video here. So we have what's called interphase. And remember, interphase is the preparatory phase prior to mitosis. You have a G1, an S, and a G2. This is where the cell replicates its intracellular machinery, its organelles, its cytoskeleton, including its filaments, tubules, etc. But most importantly, in interphase, in the S phase, we're going to be replicating the DNA, okay? Now, as we get into mitosis, actually before we enter into mitosis, there's an important checkpoint right here. And if the cell is just not good to go, if things weren't copied correctly, we'd undergo what's called cell lysis. Now, let's say everything was copied correctly. Our first phase would be prophase. In prophase, you're going to have the breakdown of the nuclear membrane. You're going to have formation or the presence of the mitotic spindle. You can see the chromosomes have condensed. And so normally I'd mentioned earlier in the lecture that when your DNA is not dividing, the DNA is kind of all over the place. But when your DNA is getting ready to divide, it condenses into what's called chromosomes. So those are those X-like structures you see. In metaphase, the hallmark of metaphase is the chromosomes are lined up in the center of the cell. You can see that the mitotic spindle has sprouted out these microtubules that attach onto that centromere, which is that little structure in the center that holds those two sister chromatids together right there. And so what's going to happen in the next phase that we have here, we're going to have anaphase, and the hallmark of anaphase is the movement of chromosomes to the opposite ends of the cell. They are being pulled by the microtubules towards that mitotic spindle that they usually call this in some books a mitotic organizing center. So it is two centrioles right here. So the chromosomes migrate towards that. In telophase, the hallmark of telophase is the reestablishment of two nuclear membranes around the corresponding chromosomes. And you can see with cytokinesis, we actually split the cytoplasm, and now we have two daughter cells that are an exact replica of the parent cell. All right, and so your next video will be on meiosis.